Vitek is back to tell us, so she focused on experimental design mainly the first time, and now we're going to hear about statistical analysis. Thanks, Olga.
Okay. Good morning. Can you hear me okay? It's working. Okay, well, thank you very much for coming back. So, as you remember, the day before yesterday, we talked about quite a bit about statistical methods and things we need to pay attention to when designing and analyzing data from quantitative experiments, right? And what we did then, we talked a lot about essentially standard statistical methods. So we talked about standard principles of experimental design, hypothesis testing, p-values, and so on. And all of this is very important, but it is somewhat insufficient for the type of data that we have been talking about for the whole week, because the type of experimental workflows which we actually have and we produce the data that we have to work with, they have a lot of specialized technological issues that we need to address as part of working with this data. And so what I would like to do this morning is to focus on the research in my group, where we use the type of techniques which we discussed on Wednesday, but now we take them to the next step so that we make this more specialized to the experimental workflows. So I would like to do two things. So first is I will spend quite some time talking about MS Stats and the purpose and the design, and I will also talk about the methods which I implemented behind that. And then also I will have the second part, which will be smaller. It's just some of the extensions which are already also up and running and are available. Okay, so first of all, the MS Stats. So this is a project which exists already now for probably 5 years or so. And it was motivated by a series of collaborations that we had, which started with DDA experiments, but they were already pretty kind of sophisticated, large-scale DDA experiments where we want to do more than have, I don't know, 3 healthy replicates and 3 disease replicates, but we had some questions which were more involved. So this is an example which was a collaboration with the Abersold lab a while ago, where our collaborator was interested in comparing protein abundances with 2 types of cell lines. So it was breast cancer cell lines, low and high invasive. And then each type of cell line was exposed to oxygen, normal oxygen or low oxygen, and then the duration of exposure was 6 hours or 24 hours. So in statistical terminology, this is called a factorial experiment, in that we have 3 types of treatment. So one is cell line type, the other is oxygen, and the third is the duration. And so different cell line growths were prepared for each of those combinations of these conditions. So it's so it is a factorial experiment, and then within each of these combinations, there were multiple replicate cell cultures. So it was a long time ago, it was glycocapture then, so for each cell culture there were multiple instances of enrichment, and for each enrichment there were multiple technical mass spectrometric replicates. So there was quite a bit of kind of different types of replication and different types of conditions. And so the question which was asked then was more complicated than just saying, okay, is there a difference between low and high invasive cell lines? So we could ask questions such as, well, is the response in time in low oxygen among the one cell line is the same as the response in time to the other cell line, right? So is this change in this cell line the same as this change in this cell line? Or like many other questions. So essentially you want to look at more complicated comparisons than just comparing 2 groups. And so this was one protein from this data set. So here the x-axis is all the combinations of all of these conditions. So this is one protein. So remember, we work on the log scale. So we will continue working on the log scale here. So this is log intensity. Different colors are the peptide ions of this protein. So these are the lines which show the average profile for the individual peptide ions, and then individual dots, these are replicates from this peptide ion in different runs. And so what we can see already that, well, the first 4 conditions is one cell line, this is another cell line. Clearly there's a difference in protein abundance between low and high invasive cell line. but we need more, right? And so if we now start comparing the effect of exposure and so on, so we see that within each condition, it's actually more complicated, right? Because the effects are more subtle. And so we see now that the individual peptide ions, they actually don't necessarily agree, right? There is some kind of interference here. And then we also have low values, which are either missing values or just noise. And so all of this, well, our kind of purpose is to integrate all of this data which pertains to this particular protein across conditions, across replicates, across spectral features, and make conclusions such as, is there a difference between the cell lines for this protein? And so when we started, our collaborator, what she was doing is actually something fairly simple. So, for example, if she wants to compare, I don't know, 24 hours hypoxia, this cell line, to this cell line, she would literally extract the subset of runs which have these conditions, take the sum of all the intensities, and then do a t-test between replicates. And so we wanted to argue back then that this is not the best way to do it for several reasons. But one of which is that if we have more than these two conditions, you know, all of these conditions, they come from the same biological context, right? And so they are somehow informative of how protein behaves. And so if we can combine all the evidence for this protein across all the conditions and all the runs, well, we will have more information about the underlying variability. We have more information about what does it mean to have an artifact of random chance versus systematic signals. And so we can hopefully provide more stable, more reproducible conclusions. And so our goal is to really take all the runs which relate to this particular biological problem and work with that while accounting for these technological issues. And so eventually this project grew out of DDA to different types of acquisitions. And so currently MS Stats works with what Brendan calls chromatography-based quantification, right? So it will be DDA, shotgun MS1, or it is SRM with or without labeled reference peptides, or it is DIA or PRM. And so, of course, these different types of acquisitions, they have very different technological properties, right, in terms of sensitivity and selectivity and so on. But in terms of the resulting data structure, which comes out of a tool such as Skyline or any other tool, actually the structure is fairly common. And so it is possible for us to propose statistical analysis, which will be kind of a general framework for all of these types of data. But then what happens is that the individual properties of the data sets vary. For example, for shotgun, you would probably have a lot of missing values and noisier features. For SRM, you would have not only peptide ions, but also transitions within peptide ions. And if you have labeled references, so now you have two sets of peaks, right? So this will be the endogenous peaks. So here, the x-axis is a run, y-axis is a log intensity, and each line is a either ion or transition. So here we have two sets of those. So these are reference peptides, and these are endogenous peptides. And so we see that the references are relatively stable, although for lower intensities there is some variability, and endogenous have some biological thing going on. And of course, DIA would have yet more different properties in that you usually have a lot more features than, say, SRM. And probably some of these features, especially among the low-intensity signal, they will have some interferences. And also the extent of missing values varies, right? And so general structure is similar, but then the details of how we go about the analysis may actually change. And so what this does currently is essentially a software tool and a statistical methodology which addresses the questions of statistical relative quantification of proteins and peptides. So what is changing, right, if I have some type of experiment? But what we also do as a kind of byproduct of that, if you want to quantify protein abundance in a single replicate on some relative scale, so the scale is not absolute quantification, but it's a scale which is comparable between the runs. So for example, if you want to draw heat maps of protein abundances across time points, right? Or if you are in a biomarker discovery and you want to have one number per protein per person, to use it as input to some machine learning tools. So we can provide this type of summarization as well. But kind of the general question is, remember the goals of statistical goals, right? So one was unsupervised discovery, comparison of averages, right? And then prediction. So this primarily focuses on this goal 2, where we compare on average over replicates what is changing between runs. So as I mentioned, we now also extend this not only to experiments which have multiple groups, but we can work with experimental designs which are pretty much of arbitrary complexity. I think by now we can be as general as we would want to be with that. So in terms of experiments, so it's chromatography-based quantification, label-free or label-based, but label-based means that we either have labeled reference peptides, or for example, if you work with SciLac, you can have super SciLac reference, which is constant, right? And we look for deviations with that, from that. And in terms of functionalities, well, we offer multiple things, so we really like to look at the data. And of course, if you work, for example, with Skyline, you do this quite a bit already. So what we do is somewhat complementary to Skyline. So our plots are maybe not, they're not as interactive as Skyline, but they provide additional type of summaries, and you can go back to Skyline, for example, to explore the details. And statistical modeling and inference, and also sample size calculation. So one of the most frequent questions that we get is, well, how many replicates do I need for the next study? And so we didn't have much time to talk about this on Wednesday, but essentially to design experiment and calculate sample size, you need to know a lot about how much variability you have in your biological system and also in the measurement workflow. So remember, if you have a lot of variability, then it's more likely to mistake changes for random artifacts. So you would need to have larger sample size to compensate for that. On the other hand, if your biological system is very highly regulated, then you don't need as many replicates. So essentially, you need to know how much variability to expect in your study. And so what we do is every time we analyze a data set, we immediately view it as a pilot study for something else. And so what we will do, we will extract variance components from this data set, and we'll say, if you have your next study, which uses this, has the same biological system, the same measurement workflow, but maybe different stresses, or maybe different scale, we can tell you what to expect from that. And the last thing is, so we feel very strongly about developing tools that people can use and not just talk about the methods. And so the MStats is implemented in R. It's free and open source. And we try to be as inclusive as we can be in that the input to MStats would be the output, for example, from Skyline. So we have a table of identified and quantified peak intensities. And so we're happy to work with Skyline, but we're also happy to work with other tools. So for example, we have a converter from MaxQuant. We have either converters or standard formats from other software tools. For example, we also can export data from SpectraNote and read it in MStats. So it is quite inclusive. And I will tell you a little bit how this works across different tools. So that's what we do. And just to kind of show you a little bit how we view the data, so this is an illustration of one protein from a particular label-free experiment, so let's say a DIA experiment. So it has two things. So the columns here in this matrix are runs, right? So they're mass spectrometry runs, which have multiple groupings among them, right? So for example, here you have multiple mass spectrometry runs which come from the same biological replicate. And this is some other mass spectrometry runs which come from the next biological replicate, and so on. And the biological replicates are grouped by some condition or stress which was applied to them. And then you have another condition, right? And again, it has subjects and it has runs within the subjects and so on. And so we have a convention in MStats that each biological replicate has to have a unique identifier. And so what we will know is that if the identifiers in this condition are different from identifiers in this condition, then we know that it's a group comparison design. Now, if we have the same identifier here and here, it means that our conditions are actually time points, or maybe it's a paired design where, if you take biopsy, like of a tumor and healthy biopsy from the same person, it would require a different type of statistical analysis, and so we can kind of recognize that by parsing the IDs. So this is essentially, so the pink color here, this is essentially the biology, right? The biological aspect of what we're interested in. And then the rows are the technological aspects. So these are different spectral features which all come from the same protein. So in DDA, it will be peptide ions. In SRM, it's transitions of peptides. So here, for example, if I have 2 peptides, 3 transitions per peptide, I will have 6 features, right? So they're all individual transitions are listed. And the same thing for DIA, so I would have fragments of peptides. And the values inside the table, they are log intensities of the peaks. And the intensity can be defined according to whatever the tools are doing, such as height of the peak at apex, full area, or maybe truncated area where you remove the tail. So whatever is appropriate here. And of course, the important thing is that there are many missing values here. And there are also potential outliers. And so to repeat what I said before, our goal is to essentially take this table and model all of the measurements in this table and make conclusions by comparing different conditions. So this is an example of the input, and I hope you will have a chance to look into that this afternoon. So even though conceptually we look at this wide format, the actual input in R is a long format. So here, one row in this table is one number in this table. And so this single number is annotated with things such as what is the protein ID, what is the peptide ID, or precursor sequence, right? Precursor charge, fragment, product charge. So if you don't have fragments or transitions, you just put it 0. Otherwise, it's the product charge. Isotope label type. So if you have heavy and light, you write that. If not, everything is light. And then these are the important things, the conditions and the biological replicates. And then the actual intensity, right? So this is the actual number here. And so this 10-column format allows us to specify all of these types of data and use the same input structure. And so now this is what is going on in terms of the overall workflow. Well, first of all, the experimental design. So remember we talked about this on Wednesday, that it is really important to think through the biological question and design the experiment in a way that will actually answer the biological question, right? And also think about how the data will be analyzed and incorporate this as part of the design. But after this is done, so maybe just also to emphasize, so this was the previous example that I showed, right? But this is another example of what we would call a complex design. So this was also a collaboration a while ago. So this was a study of childhood cancer, osteosarcoma. And so our collaborators had 29 healthy controls, which were quantified at one time point. But then they also had plasma samples on the diseased patients, and those were profiled in time. So they had some time points before the treatment at diagnosis, then during chemotherapy, before surgery, then after surgery, and they also had later time points profiled a while after surgery. And so also they can potentially ask quite complicated questions. For example, which proteins have different abundance between healthy and diseased subjects at diagnosis? Or which proteins start different, but then when time progresses, they come to the level which is comparable to the healthy subjects? Or which proteins are particularly affected by chemotherapy or by surgery? So again, there are multiple questions that you can ask. And so analyzing all of this jointly allows us to kind of better understand how much random variation we have, how much systematic variation. Okay, so this is that. But so after the experiment was designed, we collect the data. And so the first step is to work with data processing tools such as Skyline or Spectronaut or any other tool of choice. And so we export this data into R and in this 10-column format, right? And so this is where the MS-Stats work actually begins. And so we do a lot of quality control visualization. So this is a plot where here we have mass spectrometry runs. This is the log intensity of all the features in the run. So essentially we take, in this table, we take everything which comes from the run, but across all the proteins. And so the idea is that, well, we want to see if some runs have are somehow affected in a way which is not really related to the biology, but is just a technological artifact. And so what we see here, it's actually really hard to know, right? We clearly see that some conditions, so this is an experiment with 10 time points. So some time points have higher protein abundance and other proteins have lower abundance. But is it something which is really biological or is it something which is technological? And so if you only look at these plots, it's actually impossible to know because it can be both. Now, if I tell you that this is a targeted SRM experiment, which only has, I don't remember, 200 proteins, and these proteins were selected because we think that they were implicated in the process that was studied. So then it is more likely to think that this is a biological effect, right? On the other hand, if I tell you that this is a DIA experiment, which has 10,000 proteins, and we only expect a small proportion of them to change, so then you will say it's more likely that it's a technological artifact, right? So it really depends on the context. So in this particular, and it's difficult. It's really one of the most challenging things. So in this particular case, this is an experiment, time course of yeast, which was subjected to starvation progressively. This is the work from Paula Picotti's lab a while ago. And so this targeted experiment had stable isotope-labeled references, peptides, for every endogenous peptide. And so now we have these two types of peptides. So these are the references, these are the endogenous. And so now we understand, right, that the references are supposed to be stable, and they are relatively stable. But then these are the endogenous peptides, and they are changing. So now we know that it is more likely to be the actual biology. So what we do in this case, so there are some shifts in signals up and down, and there's quite a few peptides, right, corresponding to a large number of proteins. So what we do in this case in MS-Stats, we will equalize completely the boxes that correspond to the references, and then we apply the same shifts to the endogenous. And so now we can see that we really have preserved the biological effect. We accounted for some of the artifacts. So this is our kind of best practice when we work with targeted experiments, where we expect that many things can potentially change, when we have many labeled standards which we can use for normalization. However, not every experiment is like that, and in particular, DIA experiments would not be like that, right? Because we cannot spike a labeled reference for every endogenous peptide in DIA, so we have to do something else. And so the best thing that, well, there's really no good solution, right? When we have a lot of endogenous analytes, a few standards, there's really no perfect solution, but there are some solutions. So the first solution is using standard-based normalization, where we have some analytes in the mixture which are not supposed to have any biological variation, which are supposed to be constant. So there are two types of standards that we can have. One is some spiked standards. And so those will be good because we know that they're spiked in a constant concentration, but they have multiple problems. So one problem is that whatever happens to the samples before the standard is spiked, we cannot really account for that, right, with this type of standardization. And another thing is if we only have one standard, and if it happens to be in the region of the spectrum which has a lot of overlap or a lot of fluctuation, then if we attempt to normalize with respect to the standard, we may actually introduce more variation than we can correct for. So it's a little bit tricky. So just to kind of repeat it again, so the actual algorithm is what we do, we take log of the feature intensities, we subtract the median of the standard from each run, and then we add back the median of the medians just to keep things on the same scale, right? So good thing is it doesn't have biological variation. It does not depend on how many features we have on the experiment. And the bad thing is it accounts for a subset of deviation and can be noisy. And so what we have kind of proposed, and we try to do this whenever possible, is to have multiple standards, so that if one standard is somehow noisy, well, maybe we can have two, right? And so then we take the median of two standards, so that's already a little bit more robust. But do even more than that, so have some extra standards which we do not use for normalization, but use them essentially as the validation set, right? So if you have, let's say, 3 standards, you use 2 standards for normalization, and then you check what happened to the third standard. If the third standard stabilized, then it gives you an extra information that your normalization actually worked. But if your third standard got worse, then you know that whatever happened, you essentially overfit the data. And so this is a little bit of an indication of how normalization works. And so, yes, and so this would work like that. So now if you have DIA, and so now this can be, or shotgun, right? And so these are the intensities before standard normalization. After normalization, they're not all equal, right? So there's some variability, but it's not as much as before the normalization. And another approach is, which is quite common, especially in large-scale experiments, and it also comes from a long time ago from genomics and from other high-throughput experiments. So if you don't have standards, So, or if for some reason you cannot trust your standards, what else can we do? And so then we can make an assumption that in our experiment, the majority of the analytes do not change in abundance. So this will not be a good assumption for SRM, but this can be a okay assumption for DIA. When we say, okay, we have 10,000 proteins, maybe a couple hundred change, but the majority doesn't change. In which case, the median log intensity across runs should be roughly constant. And so we can do that. So for example, here it will be actually quite reasonable because after constant normalization, the medians are roughly constant with the standard. So if we wanted to if we didn't want to use the standard and equalize the medians, this could work quite similarly, right? So this is a good idea. But the problem, and so the algorithm is we subtract the median of all the feature intensities, right? So before we subtract the median of the standard, and now we subtract the median of all the features, and then we again add back the median of the median to kind of preserve the scale. So it's good because the median of everything is much more stable than a single standard, and it accounts for everything, right? Because it works with the endogenous sample, so it's a good thing. But the disadvantage is that it assumes that the majority of the endogenous proteins are not affected. And so it may or may not be the case, right? Depending on the experiment, you may actually have a lot of things. And so usually the best practice is that when we have many features and discovery experiment, we think that only a small number of proteins is affected. So that's what we usually use. And so this is just an illustration. It was a spiking experiment, or actually a titration experiment. So we know that between conditions, there's a systematic change in abundance. So then if you use median normalization, well, obviously we remove the signal, right, together with the noise. So this is not a good thing. So usually, in our experience, this is the part which requires a lot of attention, because this may be somewhat experiment dependent, right? And so it is very difficult to prescribe a single procedure, but we can say, well, these are some examples of best practice that we suggest. Okay. And the last thing also, which is possible, and this is also was borrowed from genomics and from microarrays a long time ago, is a quantile normalization, where not only we want to equalize the medians across the runs, but we want to equalize completely all the boxes. So the medians, but also 25th quantile, 75th quantile, and so on. And so this is an algorithm which essentially talks about how to do that. Well, it is obviously more stable than a single standard and also accounts for all the data processing steps. However, it is often too aggressive. So because now we will normalize away all the minor differences. So it used to be very popular in genomics. So we feel that for the type of variability that we have in proteomic experiments, we don't recommend that except for normalizing the reference peptide. So for those, we think it's actually quite reasonable. But we do not recommend this at all for the endogenous analytes. So that's the idea. Okay, and so this is the QC and normalization, and then the next important part is statistical modeling. So this is something which we put a lot of thought in, but now that it is implemented, so on your side, you don't actually need to do this modeling yourself, right? So this is the part which is automated in MS Stats. So just to remind you that we want to integrate all the information which comes from different conditions, right? And so why is it useful? So here's an example where we have 5 different conditions, right? And so so each condition has its own mean of protein abundance across replicates. And so let's say we only want to compare condition 1 and condition 2. But what we can also do is to make some assumptions regarding the variability around the mean in each of the conditions. So for example, if we can assume that the variability around the mean is the same, then even though we're interested in these 2 conditions, we can actually combine the estimates of variation across all of the conditions, and it will give us more stable estimates of variability, more degrees of freedom. This is a concept which is useful for testing and essentially helps us obtain more stable and more reproducible results. Now, you may say, okay, I don't know if the variance is the same across all the conditions. Maybe it is not. You can do other things. So, for example, you can assume that the variance is the same function of the intensity. So that, you know, on the log scale, low intensity peaks, they have more variability. High intensity peaks, they have less variability, so you can assume it's the same function and integrate it this way. So this is useful. And also, as we kind of mentioned before, right, so there are multiple layers of variation. So we have the variation in the population, we have the biological variation, and then we can take each subject and we can have multiple technical replicates from this subject. And so all of this can be integrated into one one statistical approach using linear mixed effects models. So on Wednesday, we talked about t-test. So linear mixed effects models is an extension to the t-test. So essentially, the t-test is a special case of that. It's a very simple special case. And so now when we have multiple conditions, we actually can use a similar approach, but with more conditions. And maybe what I should mention right away is that to make sure that we can accommodate different designs. So if I go back to this table, so you see that we only have one line here for conditions. So what we actually do is we create a new kind of temporary condition, which has all the combinations of all the possible treatments. So for example, in the old experiment here, so we actually had essentially one treatment which has all of these 8 different levels. And so what we will do, we'll treat it as if it was just one treatment with 8 different things, right? But then when we will ask questions about different subsets of comparisons, we will essentially extract different pairwise comparisons out of these 8 possible treatments. And so this is how we will get back to the complex questions we were asking initially. So I'll come back to that. But so for the moment, what we, just so you know, that we essentially work with one condition, but this condition can be the combination of different types of treatments. And so all of this can be integrated together in one instance of linear mixed effects model, and this will be a separate model like this per protein. So if we have 1,000 proteins, we will have 1,000 instances of these models, which we just write a loop, and we feed this model one at a time for each protein. And so going back to this table, so this is what it looks like. So we can essentially say that the intensity of the peaks on the log scale is affected by the condition from which the peak came, by the subject, so that some people have more of the protein than others, right? So we just express this natural variation, some run-to-run variation, and then also there's a feature and variation, and then some kind of unexplained deviation. And so the pink part is the biological aspect of the experimental design, so it is expressed in the pink terms, and then the yellow part is the technological aspect, so they're expressed in the yellow terms. And so this was something which we have been working with on how to design these models to extend the t-test to incorporate this kind of more complex structure. And so some time ago, a few years ago, we realized that there is one important aspect of the experiment in that there are 2 types of variation, so the biological and technological variation, which are very different in nature. So essentially, whatever happens within the run, the differences of feature intensities within the run, they have much less variation than the difference which occurs between the runs, between subjects and between conditions. And especially now as the kind of instruments improve and data processing steps improve, so the intensities of features become more and more similar in their profiles, right, in their patterns, and they are less variable than what happens within the runs. And for each run, we acquire all the features simultaneously. So this type of layout for this table in statistical terminology is called a split plot design, where we don't randomize everything completely, but we acquire all the features within the run. They have less variation. Then we move on to the next run. We acquire all the features, and so on. And so if you think about how to analyze this statistically, so in a simple case, if you had no missing values, no outliers, you can actually show that the conclusions about conditions, and this is what we're interested in, will actually depend on the summary of all the features in a run. So therefore, if we want to specify this complex model, which accounts for all of these things at once, in terms of actually quantifying each of these components, an equivalent analysis can be done in 2 steps, where first we summarize all the features in a run to have one number per protein per run, and then use this number to analyze the biological variation between conditions. And so it will be identical numerical conclusions if we have no missing values and no outliers, but this also gave us ideas on what to do if we have missing values and outliers. And so this was a really important realization for us. And so now I'm not going to kind of talk much about this part. I will just illustrate what happens in terms of the workflow. But before we get there, we need to consider again what type of data we actually have and what types of patterns of missing values and outliers we actually can have, and how things can go wrong in this model. And so this is what we do here. So remember, I was saying earlier that we try to be inclusive and we try to work with different types of data processing. So different data processing tools make different decisions, and they have their reasons for that, which result in somewhat different patterns of missing values, outliers, and intensities in this table. And we wanted to see what it means for us. So what are the implications of this for the statistical modeling? And so these are two data sets. So they are both controlled mixtures. So this data set is the DDA data set from the LFQ paper by Jorgen Cox. It was a 1-to-3 dilution. And this is a spiking experiment from one of the papers by Agnosis. And so what we decided to do is to process this data with multiple tools. And so this DDA data set was quantified with Skyline, MaxQuant, and Progenesis. And this was quantified with Skyline and Spectronaut. And so several things come out quite clearly. So the gray area here is the histogram of the log intensities of everything in this experiment. So every single value in this table across all the proteins, right? So we just like literally plot the histogram of that. And so you can see several things. So for example, the range of values which are reported is very different, right? So for example, here, Progenesis has from 10 to 30, MaxQuant has from 20 to 35. Same data set, right? So different types of values. This is not a problem because the values themselves, they don't have any meaning, right? What we want to know is how they change between conditions. And it can very well happen when, for example, you quantify the peak by the full area, right, versus truncated area. So this is not a problem, right? But it's important for us because we want to work with all of this. Now, the next thing is take a look at these vertical lines. So these are values which are rounded to 0. So they're not necessarily numerically 0, but they're very close to 0. And so you see the difference. So for example, here MaxQuant has no zeros at all. Progenesis has 665 zeros, and Skyline has 1,389 zeros, right? So it's difference between the data sets. On the other hand, missing values, MaxQuant has 38,000 missing values, and Skyline has 33 missing values, right? And Progenesis has no missing values. So it is quite different. And so if we just take this blindly, and treat everything the same, well, clearly it will not work because the intention behind these reported values is very different according to different data processing tool. And so also just the number of peaks, right? So here Skyline has 203 peaks, MaxQuant has 224 peaks, Progenesis has 56,000 peaks, right? So quite a bit of variation of everything. And one more thing, so you see that, I'm not sure if it's visible here. So there is a tail of low values in Skyline and in Progenesis, so MaxQuant doesn't have anything. So MaxQuant already has some filters. It will not report low values. And the same thing for DIA, right? So there's quite a bit of variation between Skyline and Spectronaut. So Skyline has 2,500 missing values. Spectronaut has no missing values. But then Skyline has 600 1,000 zeros, and Spectronaut has 38,000 zeros, right? And number of peaks is very different too. So this is 600,000, and here it's 2 million. So, it's kind of, it depends, especially with DIA, because we're talking about peaks, so fragment peaks, right? So the number of peaks is determined in part, for example, how many fragments per peptide you report, right? It's not like you have suddenly more proteins or anything, right? It's just different type of structure of the data. So this is all very different, but we want to make sure that we can work with all of that. And so what we decided to do is that we will essentially not take this data literally. So we will not take them as the face value, but we will try to add to this our own interpretation to understand what was the intention behind these reported values. And so here is this vertical dashed line that we have. And essentially what we're saying is that below this vertical line, we do not trust the numbers. Call them 0, call them an A, call them small values, whatever you want to call them, we do not trust them. So now this vertical line is determined somewhat empirically, so there's really no deep science behind that. So what we decided to do is we took, I think, 99th quantile, so on the right tail, such that only 1% of peaks is above that. Then we took the median and we flipped this on the other side of the median. So because we just think that the right tail is more stable than the left tail. And so this is roughly our kind of estimate of the cutoff. So it's dataset specific cutoff, but it's based on the entire experiment. And so essentially we say that everything below this line we cannot trust. So we treat them as missing values, but they're not just randomly missing values, they're actually informative missing values. So we think that they're missing because they're low abundant, because some of them have either small values or zero values. So an exception is Skyline. At least for the moment, this is the only tool which we know which distinguishes missing values at random from missing values due to low abundance. So for example, if you have a scheduled SRM and your peak moved outside of the scheduling window, so the peak is heavily truncated, you cannot determine its area. So then you know that it was there, but it's not quantified. So then it's an NA. But then when the peak is not there, so you essentially integrate the noise area in the expected region of the peak, so it's a small value. So that we know. But for example, MaxQuant treats everything as NA. So then our interpretation is that every missing value is missing for reasons of low abundance, just because it's more frequent. So we cannot distinguish that. And the same thing with other tools. So except for the fact that for these truncated peaks, we leave them as NA, we don't do anything with them. For the peaks which are missing for reasons of low abundance, we actually treat them as what in statistical language is called censored values. So they're missing, but they're informative because we know that they're small. We don't know exactly the value, but we know that they're small values. And so now this informs our overall workflow. So what I said earlier is we can do it in 2 steps. First, we summarize all the feature intensities per protein per run. And now also what we do, we need to incorporate in the summarization the information on the missing values and outliers. And so now we have this table, so we only focus on the technological part. We kind of ignore for the moment which run came from which condition and all of that. And now we replace the missing values with our own missing values, right? So now we have NA censored according to our definition, and we add NA random in the case of Skyline if it was a missing peak. So that's what we do. And then from this point on, you can actually focus on this as a 2-way analysis of variance. So you just look at this as a table where you say, okay, each feature intensity is determined by, well, which feature it comes from, because different features have different overall intensity. And then from which run they came, because different runs have more or less variability. And when we quantify the feature levels and the run levels, we incorporate this censoring mechanism. So this is using maximum likelihood procedure, if you're familiar with that, but this is an extremely standard technique. So it is frequently used, for example, in clinical trials. You know, when you follow up a person, like the survival, and then the person moved away and you lost track of the person. So you do not know exactly how long the person survived, but you know that the person survived long enough, right? And so this is also used. So it's a very similar technique here. And so we have that. And in principle, this is enough. However, because it's using this maximum likelihood procedure, it is very sensitive to outliers. So it will take care of the missing values, but it will be affected poorly by outliers. And so there are ways of kind of using robust estimation, which looks at both censoring and outliers, but we find that it's not very stable numerically. So what we decided to do is to simplify this a little bit. So what we will do, we will actually use this censoring. We will predict the values which are missing from this, and then we'll have a second layer of analysis using a robust method to account for outliers as well. And as the result of that, we essentially obtain this summary of one number per protein per run. And from this point, we get back to the biological information, and then we can use kind of fairly standard statistical methods depending on what was the experimental design. So that's what we do. And so now, remember I was saying we still need to figure out which pairs of conditions are changing or some linear combinations of conditions. And so from this point, this is where we will write a linear combination. I want to know if the average of 6 and 24 hours of exposure in normoxia, the same as the average of 6 and 24 hours exposure in hypoxia, right? And so you can write linear combinations of all of these conditions to get to the comparisons of interest. And this is very easy to do. Okay, and so finally, so this is something that you would specify as part of the analysis because we do not know which comparisons you are interested in, right? So this is the only thing that you need to specify. Well, and after that, the output is in form of the volcano plot, which we can already looked at a little bit, right? So this is the estimates of log fold change. These are the estimates of p-value, adjusted for false discovery rate, and minus log, just so that the kind of small p-values actually have high values on the negative log scale, right? And so we can also have the fold change cutoff to have what we call practical significance, but it's important to have the statistical significance cutoff. So the cutoff here corresponds to the false discovery rate cutoff. And just a comment here, because I know there has been a lot of discussion of false discovery rate for identification, right? So when we work with all the protein level, peptide level, and so on. And so suddenly here I talk about false discovery rate for quantification. I just want to clarify that these are two completely different things. So what happens, I think, in this analysis, for better or for worse, is that especially if we work with experiments such as DIA or DDA, where we need to characterize our confidence in the identification, so we would use some techniques to quantify the uncertainty, right? We will report Q-value, we will use Q-value filtering. We would use false discovery rate as a guideline for which cutoffs to use, but that's pretty much it, right? So at that stage, the Q-value filtering is used as a score primarily, right, which has some probabilistic interpretation. And when we go here, we essentially start over. And so now the false discovery rate kind of refers to what is the proportion of false positive changes I have in the collection of the reported changes. And so this false discovery rate is essentially conditional on whatever has been done with the identification. So it does not propagate the error rates in the identification into the error rates of quantification, for better or for worse. So this is the state of the art. I'm not aware of any generally used. There's some kind of research level methods, but I'm not aware of any kind of daily used methods which would propagate the uncertainty in the identification into the uncertainty of quantification. So this is just to keep in mind. So in fact, even though we control false discovery rate here, we may actually have more uncertainty which comes from the false discovery in the identification. And so that's what we can use. So we can use the cutoffs. But another question which I often get is, well, I get some p-values, I do false discovery rate, nothing is significant. Oh my God, my experiment doesn't work. So what can I do? So especially if these are discovery experiments, which are hypothesis generation experiments, you can use false discovery rate cutoff to establish your list. But what you can also do is just rank your proteins by p-values and then say, I choose to follow up on that many top proteins, regardless of the false discovery rate. And I think it's also a fair way to do it if it is a hypothesis generation experiment. Okay, so this is what we do. And so let me now show you how it works in terms of the summarization and hypothesis testing, right? So this is one spike-in data set, so it's a DDA spike-in data set. So 4 conditions, and this is one protein. So spike-in experiment had some background proteins which do not change and some spiked proteins which change in abundance. So here we have the gray lines. These are the actual peptide ions of this protein. And the color lines, these are different summarizations. So the green line is the summarization which is often used. So when you take the intensities of the fragments or ions of a protein on the original scale, not log transformed, take the sum and then take the log. Right, so this is very commonly used, and I think Skyline is using that. And another approach is, in yellow color here, it's kind of hidden, is using this big linear model, so which doesn't take care of missing values and doesn't take care of outliers. Then another orange color is the robust approach, which looks at the outliers but not at missing values. We just try to kind of understand the relative contribution of working with missing values or outliers. And then the red color is the proposed approach. So what you see here is that the summarization with the log of sum, well, it's above all the gray lines, right? Because it takes the sum of the intensities. Of course, it will be higher, right? But other than that, everything is kind of looks nice and good. So the blue dashed line, this is our cutoff, where we say below this cutoff, we don't trust the values. So here, all the measurements are nice, right, and clean. And when everything is good, right? So every method does pretty much the same. Here, comparing two conditions, true, false change is 1. Everybody's very close. No differential abundance, so no problem, right? So this is good. Now, here is the case where we have some outliers among low-intensity features. So when we have low-intensity features that are not quantified correctly, log of sum is not really affected because small values, it doesn't really matter, right? It's really the big peaks which affect the log of sum. But linear models would be compromised right away, right? But then the other methods are kind of the same. But now when we have an outlier in high-intensity peaks, well, then the situation is different because now log of sum is actually very much affected by this, and also the linear models are affected. And so here we compare condition 4 to condition 1. The true-false change is 1, and log of sum will have 3.5, so it will exaggerate the difference between conditions. And the robust method and the proposed method, they're actually identical because there are no missing values, right? So then they are identical here. And they will do better than the linear model. But real life is sometimes like that, right? And so these are the cases where you have most differences. And so here we see we have some outliers among high-intensity features. We have some missing values, which we just display here with zeros, which are below our kind of trustworthiness cutoff. Linear model is affected by the missing values. Log of sum is affected by outliers in high intensity. So linear model, so here we compare condition 1 to condition 2. True-false change is 7.5. Linear model thinks it's 29. Log of sum thinks it's 1, so it's all over the place, right? But when we take care of the of both missings and outliers simultaneously, then we're much closer to the true value. So that's kind of the idea. Okay, and so then just to kind of show briefly that we can extend the same thing if we have labeled references, so that works kind of the same. And finally, as I mentioned in the beginning, we work on the experimental design, so we extract variance components. So the good thing about this big linear mixed effects models is that you explicitly quantify the contribution from different sources of variation, such as biological variation, technical variation, if your design allows that. And so then we can really project the required sample size from that. And so we would produce a plot that looks like that. So it's very clear, right, that if you want to detect smaller fold change, you need more replicates, right? So this is clear. But what sometimes people forget is that the number of expected differentially abundant proteins also affects the sample size. So the fewer true changes we have, the larger sample size we need. And this is sometimes counterintuitive, but just think about that, right? So if we have a targeted experiment with 20 proteins, and we target these proteins because we know they're all changing, well, everything is a true change, so there's no false discoveries, right? So there's no problem, right? On the other hand, if we have a DIA experiment with 10,000 proteins and only small numbers changing, there is a lot more opportunity for false discoveries. And because we need to protect ourselves against false discoveries, we need to work harder. And in this case, work harder means having a larger sample size. So these global large-scale experiments have a price, right? So it's kind of nice. You say, okay, we can quantify many things, but if you are not really interested in those other things, well, this is the extra price you have to pay by essentially extracting more things than you need. And so this is also part of consideration, you know. So even though you can potentially detect many things, do you really need all of them? So if you can consider a subset, it can help you at the stage of quantification because it will reduce your multiple testing. Now, how do you decide which ones you want to use? Well, it's not always easy, but you can at least have some filtering, for example, based on, you know, I don't know, cellular versus extracellular proteins, right? Or proteins which have no functional annotation, or proteins, you know. So if you don't need them, don't use them here because it can help you with the sample size. At least at the stage where you really want to report error rates. Later you can kind of look at this as well, but this will be part of your exploration and hypothesis generation. Okay, so let me show you evaluation now just a little bit. So we are interested in comparing the methods, but we're also interested to be robust across data processing tools. So here is one spike in experiment. So we partition this into true-false changes. So proteins were spiked in different concentrations, so each row corresponds to different fold change in the pairwise comparisons. And each column is one of these 4 methods, right? So here it's log of sum. This is just linear model, no outliers, no missings. So this is only handling outliers. This is handling outliers and missing values together. And so the green color is sensitivity, so darker is better, and blue color is specificity, so again, darker is better. And so you see that in this case, it's pretty clear that for this data set, the proposed approach is much more sensitive without compromising the specificity. So this is very nice. And so now we try to extend this to different data processing tools. So all of this is DDA experiments. So this was this data set processed with skyline max quantum progenesis. And then we have 2 more data sets also processed with 3 or 4 tools. And so it's not always perfect. It depends on the type of variability and the type of measurements. But except with some really small kind of isolated cases consistently, the left column performs consistently well. So it gives us confidence that across different data processing tools, it's quite robust. So the same thing for DIA. So here is one case where it's not as good as linear model, but linear model generally doesn't perform well. So I don't know what is going on. But generally, in most cases, the left column performs consistently well for different types of data processing tools. So this is good, but what I was really interested in particular, is to understand how this affects our downstream, how the choices of data processing tools affect our downstream conclusions. And so what we looked at, we looked at different spike-in mixtures. So these are 3 DDA spike-in mixtures for which we know what proteins and which comparisons have true changes. And we looked at the intersection of detection of differentially abundant proteins among the true changes among data processing tools. So here we see that if we use, so let's say here we use the log of sum quantification. So if we process these data sets with 3 tools, only 11 proteins are found by all the tools. However, if we kind of do this kind of more kind of extra modeling, right? So then the intersection between processing and conclusions of different tools is better. And it turns out to be across the board. So for log of sum, we also go for this data set, we go from 29 to 32 changes. Here we also increase from 123 to 146. So essentially what it means is that we don't take our values kind of on the face value, right? And if we really try to understand what was the intention behind these values and we model them more appropriately, we can mitigate somewhat the difference in decisions which these different tools make. So to me, it's a really good news because it means that we can improve the reproducibility of the results regardless of the workflow that we use downstream. And the same thing for DIA. So also the proposed approach will increase the overlap for this spike in mixtures. And the same thing for estimates of log fold change. So here we take all the DDA mixtures, all the DIA mixtures, and we also had a whole collection of SRM mixtures. For SRM, and this is the true log fold change, and this is the reported log fold change. And so processed with different tools. For SRM, there's not much difference because, well, usually the data are much cleaner. So there are fewer outliers, fewer missing values. But for DIA and DDA, you can see that in terms of estimates of fold changes also, it does improve. So we think it has a good news. And so, as I mentioned earlier, so MStats is open source, implemented in R. And so we have multiple ways of accessing that. So this is the MStats website, which is probably the best resource. So it is also available on Bioconductor, but I don't actually recommend to download the Bioconductor version because Bioconductor only gets updated twice a year. But we have the daily version, which gets updated more frequently. So MStats Daily is a much better way to go. And so it is also available as Skyline external tool, which is very exciting for us. So part of the analysis can be done entirely through the Skyline graphical user interface. Unfortunately, there are some limitations to what we can do with that, so you get more options with the R command line, but that's a option as well. So yeah, this is quite nice for us. So that kind of finishes the MStats part. I have other things to share with you, but maybe first let's see if you have any questions. Yeah.
What I saw so far, for p-value adjustment, you're using Benjamin-Hochberg procedure.
Yes.
Was there any reasons why you didn't include the q-value, like after a story?
Yes, there is a reason. So you're right. So Benjamin-Hochberg was the first procedure, and it is the most conservative procedure in that it assumes that the worst case scenario, it assumes that nothing is differentially abundant, right? And so it adjusts in that case. So there are alternatives such as story procedure and efference procedure, which actually estimate the proportion of things which are not changing and only controls FDR among those. So because the estimated proportion of changes is smaller, so it allows you to be less conservative, right? And so this is a good thing. The problem is that these methods, they have some numerical calculation steps. So for example, story method fits a two-group mixture to the p-values. Efference method fits a two-group mixture on the transformation of the test statistics. And so the problem is that these two-group mixtures, depending on your data set, they can be very difficult to fit. And their estimates of those mixtures can be quite unstable. And so it really affects your estimates of Q values, depending on how many true changes you have and what's the distribution of the p-values. Benjamin-Hochberg, on the other hand, doesn't have any of that. It's a very simple simple transformation. So we find that it's numerically much more stable. And because our goal is to provide a robust tool, maybe not like the best, but like, the tool which goes like across most data sets, we felt like it's a better way to go. However, as part of the output, we output both unadjusted p-values and Benjamin-Hochberg p-values. So it's a one-liner in R to use some other methods, so you can totally do that.
One question, you mentioned the quantile normalization as aggressive, but I would like to ask you for your opinion about the SILAC data sets and quantile normalization. Is it also not recommended? How would you comment?
Quantile normalization doesn't depend on the technology, right? It really depends on the pattern of changes, right? So it really depends how those quantiles kind of fall together, right? But essentially you require a lot more reproducibility between the runs. So reproducing the median signal is one thing, but reproducing the maximum and the 99th quantile and the 98th quantile. So you make a lot more changes to your data. And so the more you manipulate the data, the more chances you have to, reduce the biological signal. So usually my intention is to do what's needed, but as little as possible, right? And so we try to go at most with the equalizing medians to avoid aggressive manipulation of the data. And that doesn't really matter if you have SILAC or whatever else. So I don't have any kind of theoretical things for that besides just empirical evidence that sometimes it reduces the actual biological signals too much. Yeah.
Can I ask one? So many other softwares are implementing for global normalization TIC instead of median. Do you have a comment about that? I think I found in the past that median was a bit more aggressive than TIC, but do you think it? Well, have you looked at this?
Not recently. So essentially what you are doing, you are assuming that something should be constant, right? So is it better to assume that the total ion current is constant or all the peak intensities are constant, right? So it depends what you add to this by considering TIC, right, as compared to the peak intensities. So you add essentially noise, right, or peaks that you don't need. Now, is it a good thing to assume that those things are constant? Well, maybe if your noise is constant, right? So I think especially in DIA experiments when you have so many peaks, I don't think it really matters. I think in DDA it would matter more because the number of peaks is smaller. But once you get to like as many peaks as we get now, I don't think it really makes a difference. Yeah.
So about the definition of features in DIA, data analysis, would you recommend to consider a single feature, a single fragment ion, or the sum of all the fragment ions relative to a certain precursor?
Individual fragments, so that's what we do. So yeah, if you have 3 peptides and 5 fragments per peptide, you have 15 features. Yeah, and the reason is that when you take the sum, well, all the problems which I just discussed with the sum, if you sum at the peptide level, you will have the same problems, right? So if you have outlier among the highest peak, right, or missing values. So What we assume here, and this is maybe a strong assumption in some cases, is that all the peptides and all the fragments, they're all behaving the same way for the protein, right? And therefore, it makes sense to summarize them and make protein-level conclusions. If you have a peptide which you think may behave differently, like if you're interested in PTMs, for example, right? So then different peptides may not represent the same quantitative trends, right? So then you just shouldn't be doing protein inference, right? So you can do peptide inference, right? And that's fine. So in MS Stats, we just have a trick. Instead of the protein name, you replace it with a peptide name. And then all the conclusions are made at the peptide level and not at the protein level. And then you can kind of potentially summarize each peptide separately. The problem is that then you increase your number of comparisons, right? And so at the multiple testing stage, you will be penalized quite a bit. So I wouldn't do it per peptide unless you have good reasons to believe that some peptides really should not represent the protein. Yeah, but other than that, so yeah, well, we consider that all the features are expected to have exactly the same profile, regardless of which peptide they came from. Okay, so let me just take the remaining time to tell you about 2 additional extensions to MS Stats that we have recently. And so the first extension was actually motivated primarily by DIA experiments, in fact. So a lot of people now use DIA to kind of in the sense of a targeted assay, right? Where we have a panel of analytes which we want to quantify consistently in different types of conditions, and so we will consistently profile those. And so if that's your workflow and you always work on the same analyte, so eventually you want to know what are the characteristics of these assays, right? So you want to know what is the dynamic range, you want to know what are the limits of blank, limits of detection, limits of quantification. So you can do it for this purpose, but our motivation was slightly different. In the beginning, we looked at different DIA workflows, and we wanted to know which type of DIA workflow gives us better sensitivity and so on. So that you can also determine by characterizing the assays in terms of limit of detection and limits of quantification. And so usually the way you do this is you have a titration, right, of analytes. So either you spike them or you titrate the mixture, right? And you make a plot where you look at the signal versus true concentration. You make the points, and then you fit a straight line. And you probably all have seen something like that, right? So we draw a straight line. We maybe have some intercept and some slope, right? And so this particular example came from the Open SWATH from Hannes Rost paper a while ago. And so we want to be able to quantify the background, quantify the slope, which has some interpretation. And usually what people would report is correlation or R-square, right? And in this case, it looks good. So it looks quite nice. And I have seen, I know you probably have seen thousands of plots like that, right, with a straight line. And so I would actually like to argue that this is not a good plot, and this is not a good way to characterize the assays. So first, let me just tell you a few words why it's not. Oh, before that. So what do we want to do from this plot? So we want to determine things such as limit of blank and limit of detection. And there are actually multiple definitions of that, but there are some of them which are standard, and so that's what we use here. So limit of blank, what we can do, we can think of the upper limit of prediction interval of blank samples and see how it intersects this curve that we fit. So limit of blank is here. So if this is the average signal from the blank samples, and this is their prediction interval, so this is the range where we can expect new data points. So then the limit of blank would be the point where our curve fit intersects the upper prediction limit of the blank samples. And then limit of detection now considers not only the prediction interval for the noise, but also the prediction interval for the actual analyte. And so limit of detection will be where the lower prediction interval of the straight line intersects the higher bound of prediction interval of the blank samples. So just to also point out that prediction interval is different from the confidence interval, right? So confidence interval characterizes how confident we are in the curve itself. Prediction interval is what is the range of values we can expect in the future from this curve. And so these are the two quantities we want to make. And so, yeah, it used both to characterize the assays and compare the technologies. Okay, so now if we take this curve, so I would like to argue that even in this particular case, using this curve is not a good idea. And so here is why. So here I'm looking at all the concentrations, right? I fit straight line. So, well, okay, so if we look here, the intercept is negative. So what does it mean, right? When the concentration is 0, so the analyte is absent, we have negative signal. Doesn't make much sense, right? Well, okay, we can kind of ignore this, right? Now, well, I can also try to add a smooth curve, right, here to see, well, how linear things are. So here I'm adding a red smooth curve. Well, it kind of dips here, but hey, who cares, right? And overall, it's kind of all linear, right? So it doesn't matter. But so now what happens is that if I decide to remove this highest concentration, and I make exactly the same plot, but without this concentration, and I use the smooth curve, so suddenly I see that it dips a little bit more. So essentially, we can make our plots look as linear as we would like by adding higher concentrations, which is a problem because what we're actually interested in is low values, right? And it's very easy to mislead us by adding high concentrations, and it looks like the fit is good when, in fact, the region where we're interested in is non-linear at all. So why is it a problem? It's a problem because if you do targeted SRM assays, you can probably fine-tune the true concentrations in dilution such that you are never in this particular region, you are always only in the region of linear concentration, so that the linear curve is actually reasonable. But if we want to characterize the DIA assays, which have so many analytes, so we have to make titrations or spikes of that many different peptides. It is impossible, or at least not very practical, to kind of fine-tune the true concentration. So what we will have often is some concentrations which are so low in the lower end of the dynamic range that the signal will flatten out. And so here's another example from that. So it kind of looks linear, but when you zoom into the lower range, you see that it's not linear at all. And this is what happens when you have to characterize essentially highly multiplex many assays. So you would be in these situations. And so the question is it a problem? And it turns out that it is quite a bit of a problem because it would affect our curve fit, and it would affect the definitions of limit of detection and limits of quantification. So what people do to correct that sometimes? So, well, we can use some robust fits, which don't really make a big difference. We can fit some logistic curve, like S-shaped curve, which look kind of like what we would expect, right, from the calibration, but they have many problems themselves, so they don't fit. Like here, I have some logistic curve examples that don't quite fit the data. And again, if we want to apply it on a large scale, numerically, they will not converge if we don't have this kind of flex point. So it's a problem. So we started working with these assays primarily to decide which workflows work better, but then we realized that for kind of large-scale use of DIA, we need to do something better than that. And so what we decided to do is this. So we went back to the definition of what actually happens in the instrument, right? And so this is the canonical calibration curve, which says that in the blank samples or in some very low abundances, the signal is essentially flat. So we only see the noise. And starting from some point, we start having a linear relationship between the true concentrations and the signals. And so this flex point is unknown, right? So this is a parameter of interest together with the slope and the intercept of the curve. And so it turns out that this kind of what we call it a hockey stick form can be written like this. So it's a nonlinear model, but very simple model. So it's flat up to a certain point, and then it's a straight line after this point. But the problem now is that we have an extra parameter, which is also unknown. And so the uncertainty in this flex point is a really big part of the analysis. And so what we do then, we would fit this type of hockey stick curve, but we do it many times. So what we'll actually do, we will, to characterize the uncertainty, that we will resample the data. So the statistical procedure is called bootstrap. So when we sample with replacement some of the data points from this protein, we feed this curve. We sample again with replacement, we feed this curve again, and we do it many times, maybe 1,000 times. And so here you see we have the gray lines which correspond to this 1,000 iterations of fitting this curve to resample data. And so if the data are resampled in a way that you don't have this flex point, it's only straight, you fit a straight line. If you have a flex point, you fit this. And so there are these multiple curves which you obtain, and then you average the results of these curves across all the iterations. And so what you obtain is that it becomes a straight line if the peptide is entirely within the linear regime of the dynamic range, but it will be a non-linear curve if the peptide is flattening out like that. And this kind of flex point is not the result of the actual kind of flattening of the signal. It's more the result of our uncertainty in the change point of the calibration curve. And so this is also now available. Oh, and just to show, by simulation, we have a whole collection of data sets where we show that this essentially matters quite a bit. And the main effect of that is that our limits of detection and limits of blank, they're actually much higher, not much higher, but higher than what we would expect just from fitting a straight line. And this is really important because the straight line, if it doesn't account for this, it gives us too much optimism. We can think that we can detect things much lower than what we actually have. And so that's a really important point. So it kind of brings back a little bit the expectations from these methods. And it's important because if you have your expectations right, you will know how to use these technologies more effectively. And so it's part of MSStat, so you can download this, and it also has user manual and so on. So this is one extension. And so the other extension that we have, which is also fully available, has something to do with running large-scale experiments, but not in terms of the number of analytes, but in terms of number of samples. So as we talked already before several times, that number of replicates really matters. And so if we start running experiments which have more larger scale in terms of the number of replicates, then we need to make sure that our measurement system is stable, right? It's more likely that something undesirable will happen if we run the experiment for a longer time. And so we have considered a statistical methods which now focuses not on the biological samples, but on the standard and calibration samples. And how can we make sure that during the entire time of the experiment, there are no problems? Or if there are problems, how can we detect these problems? And so in statistical language, this area is called statistical process control. And it's actually quite a vast literature by now, and it is very much used in industry, in different areas of manufacturing. What is different for us is that unlike in other industries where they measure a couple things and they monitor them in time, we have a lot of things, right? So if we have DIA experiment, we can have standard samples which we interleave between the endogenous samples. We can have potentially 1,000 peptides which we can quantify in these samples. Each peptide can have all kinds of readouts, such as are retention time stable, are peak areas stable, peak intensity, peak asymmetry, you know. So all kinds of readouts you can have potentially from that. And that's not all, because now different problems can also occur. So maybe you can have some individual spikes which are like out of control. Maybe you can have shifts in the mean retention time. Maybe you have shifts in the variability of retention time. And so all of these things would require different diagnostics. So it's essentially a very highly dimensional multivariate system, which is very different from what most industries experience. And so we really need to be able to work with these very high dimensional systems. So this is what is currently used, and it's actually implemented in Panorama as well, and also in the external tool to Skyline. So you can, for example, here monitor through the time, in this case, peak area of a particular peptide, which is a standard peptide. And this peptide can be spiked in your endogenous samples, or it can be just in a standard mixture, which you profile at the same time. And so what you can do is to take some number of runs, which you view as good quality runs because you think they went well, and you can estimate the extent of variability of peak area of this peptide in these runs, and it can define the limits within which you think that the peptide performs well. And so if the signal extends beyond these limits, you say that this is a problem, right? You flag this as an issue. Or you can use the z-score, which is essentially the peak area divided by the standard deviation of the peak area. Now it's on the scaled it's scaled, but it's the same profile, right, and the same type of things. So this is useful, but it only helps you diagnose the shifts in the actual peak area. What about shifts in variability? So turns out that there are some other summaries, so it's called moving range chart. So it summarizes our peak intensities in a different way, but now it monitors shifts in variability as opposed to shifts in the mean. And so again, from the guide set, you can estimate what would be the appropriate bounds. And now you can see that, so this was actually a simulated example. We spiked some kind of problems, and now these problems resulted in the increased variability. So you can also diagnose the variability. But that's not everything, because now these are the kind of big changes in the mean. But sometimes what we actually see is not the big change, but subtle drifts, which are small, each of them, but together they kind of contribute to the system drifting away from the optimal performance. And so here, for example, we have a peptide which increasingly drifts. So here peak area keeps increasing, but increases kind of in a subtle enough way that it still stays within our limits, so we cannot detect this. And the same thing for variability. It increases little enough that we cannot quite see it. However, if we use a different summarization, so this is also a fairly standard way to summarize the data to detect small drifts, we can also have a chart like that, and also use guide set to establish the controls to see that starting from this point, we actually have a drift in the peak area. And we can do the same thing for variation. And what we can also do with this type of summaries is to not only say, okay, now we have a problem, but we can also say we can estimate the time point where the problem started. And so this is also important because it allows you kind of to give an objective way to say, starting from this run, the drift occurred. And this is important because, you can look at these charts one by one, but if you have 10,000 peptides and many metrics for them, you need to be able to do this automatically, right, and highlight problems in a way that you can easily interpret. And so this is what we do now. So this is also part of MS Stats, and usually MS Stats is primarily command line based, but this particular tool, we actually have a graphical user interface with a Shiny app, so you can upload your QC data and do visual interpretation of that. So we have this, what we call a decision map, where for different metrics, so here we have peak area and retention time. This is for the mean, and this is for the variability. Across all the peptides, we essentially just say blue is good, yellow is problem, like warning, and red is a problem. And so you can just take a look at this heat map, which summarizes all of the metrics and all of the peptides to say, okay, go or no go. Now, if it's a warning or no go, then you can dig a little bit deeper into that. So here we have two types of view. So this is a type of problem view. So here we have the run on the x-axis, and different lines highlight the proportion of peptides which have problems. For example, increase in the mean signal, decrease in the mean signal, increase in variation, or decrease in variation. And so you see that here, the first few runs, no problem. So now this particular, this is the proportion. Up to 50% of peptides have an increase in the mean signal. And the red dots here pinpoint the time points where the problem started for the individual peptide. So you can kind of see from which one it's a problem. So this is for retention time. This is for peak area. So clearly peak area started to drift. And so this is the peptide view. So this is kind of the opposite view of that. So here we summarize over peptides. Here we summarize over metrics. So this is the proportion of metrics where there is a problem. So you want it to be close to the center. And so peptides which have colored areas outside of the center, these are the peptides which tend to have problems. And color indicates which type of problem. So you can also kind of see quickly which peptides create problems. And if you do see problems, so then you can go to these individual charts and kind of dig into the individual peptide and try to understand what's going on. So this is also, we can, so just to summarize, we can have detections of large aberrations, small sustained drifts, monitor multiple peptides, and we can show that the more peptides we monitor, the more problems we can detect, right? But at the same time, we can also produce rules which will control for the kind of high dimensionality and multiplicity of this. And so this actually is published, so you can have the description of all the methods in the manuscript. And so this is the graphical user interface that I had mentioned. Okay, so I want to make sure that I acknowledge those who actually did this work. So Mina Choi, who is standing in the back there, she is the main person behind MS Stats. She has been leading this effort for a number of years now. So she has developed the main methods behind MS Stats. Ting, who is sitting right there, so she's also a contributor to MS Stats, and she is involved in a number of MS Stats projects. Cyril was leading the LOD/LOQ project. Sung Heng is also a active maintainer of MS Stats. And Eralp, he's not here, but he was developing the MS Stats QC together with Sarah, who is here. And a lot of this work really relied on collaborations with many groups who helped with the data, but also testing what we did, or developers of data processing tools such as Skyline and Brandon in particular, to help integrate our workflows, create converters or external tools. So there's quite a lot of people who were involved in different aspects of that. Okay, sure. Thank you.
Thanks very much, Olga. Are there any further questions now? So I have one about the missing value. So in your proposed method, I guess a hard case to deal with is when there's missing values completely missing from an experimental group. Do you handle this? Or?
Oh, yes. So I forgot to mention. So, right. So, well, first of all, if the missing values are scattered kind of randomly, right, like it's a really like poor quality feature, you can just remove it up front, right? If we have some individual isolated missing values, we do the imputation. Now, if you have one condition where the peptide or protein is completely gone, but it's present in another condition. So you can't really do any statistics with that. So what we do, we just output log fold change infinity and p-value 0. So when you have log fold change infinity, you know that it's qualitative, it's not quantitative, but we want to make sure it makes it to the top of your list. Mm-hmm. And so we just essentially complement this with a qualitative step. We just say it's log fold change infinity.
Is there some way to handle that? So presumably if you have more replicates in both groups and missing values completely in one and present values completely in the other, so as you grow the number of replicates, it seems less likely that will have happened by chance. So is there any way to treat that part statistically?
So if you have at least one replicate in the group, then you're good, right? Because you can borrow the variation from the other group, right? Yeah. And this single replicate will give you the estimate of, well, it won't be very good, right? Because it may be a variable, right? But at least you have some numbers to deal with. So you can definitely do it.
I mean, rather the case like where, so let's say you had a case with 3 replicates and 3 replicates, and in one side there was completely missing values and complete values on the other side, and now you have 10 replicates and 10 replicates. Mm-hmm. Then you have all missing values on one side and all complete values on the other side. So it seems like you should be more confident that this presence-absence result is more likely to be true in the 10 case than the 3 case. So is there a statistic that sort of
not really
captures that?
Not really, no. Because you still don't know what the actual fold change is, right?
Okay, but is there a way to say that arrangement of values wouldn't have happened by chance, for example? Not really. Okay.
Maybe, but you know, I would rather say it's qualitative because we don't see it, right? But it's good enough to follow up on that. So, for example, if you want to do things like biomarker discovery and use this as input to prediction rules, mm-hmm. you don't have the actual value to put in for the group which you have. So that's and you would need to have a value per person, right? So you would not be able to proceed with this unless you start making up values like zeros for each person, but right? So it's important to highlight that, right? So you don't want it to make it look similar to the proteins which actually have the data, because it is a different case. And so that was just the kind of the decision we made, that we want to make sure it's reported, but we also want to make sure people know that it's qualitative.
Yeah. Great. Anything else for Olga right now? Okay, great. Otherwise, we'll go to coffee and come back at 11. Thanks very much, Olga.